Welcome to all. In this class, we study about glycolysis. Glycolysis means glucose lysis, which means the breakdown of glucose. It is defined as the degradation of glucose into two molecules of pyruvate or lactate by sequence of enzymes, catalyzed enzymes, and finally to produce ATP. It is a catabolic pathway. It is the essential pathway of carbohydrate metabolism because it can synthesize the energy for us for working. If a glucose is degraded into pyruvate, then it is called an aerobic glycolysis. It is in the presence of oxygen that is occur, and if glucose is degraded to lactate, then it is anaerobic glycolysis, which means the final product we get lactate, that is anaerobic glycolysis. It is occur in absence of oxygen. It is also called as M10-Mayerhof pathway because this glycolytic pathway is elucidated by M10-Mayerhof in 1940. Next, we see about the site of this glycolysis. Enzymes of glycolysis are present in cytosol or cytoplasm of every cell. So that is occur inside of cytoplasm of cell. Then sources of glucose. Wherever we get the glucose for the glycolysis, dietary glucose formed from the digestion of dietary carbohydrates, then it enter into liver through portal vein system after its absorption from the intestine. So the dietary carbohydrate rich in glucose. After digestion, that is reached into liver by the portal system. The liver distributes glucose to all other organs of the body, which means that every cell gets the glucose from the liver. Entry of glucose into the cells. Glucose enters cells by facilitated transport. There is a specialized transport system that transport the glucose into cells. First, the liver glucose enters liver by facilitated diffusion. It is an insulin independent transport mechanism for the transport glucose across liver cells. Next one, extrahepatic tissue, which means the other organs other than the liver organs. The glucose enter adipocytes, erythrocytes, brain, and skeletal muscle by facilitated transport involving carrier molecule. The transport of glucose across the membrane of these tissues by carrier is independent on insulin. Next, we see about the reaction steps of glycolysis. The totally 10 steps are present in this pathway that is divided into three phases. First one, energy investment phase. Next one is splitting phase. Then third one, energy generation phase. Energy investment phase, splitting phase, energy generation phase. We see about the first phase, energy investment phase, in which the energy is invested, which means these phase require energy for the carried out these reactions. This phase there are three steps. First, the glucose is phosphorylated, which means the phosphate group is added in glucose and produce glucose 6 phosphate by the enzyme hexokinase or glucokinase. Both are the isoenzymes, which means the same molecular structure but different spatial organization. Then the next step, the glucose 6 phosphate undergoes isomerization to give fructose 6 phosphate. The fructose is the isomers of glucose. So this conversion is carried out by the enzyme phosphohexose isomerase, in which the Mg2 plus is act as cofactor. Next step, fructose 6-phosphate is phosphorylated to fructose 1,6-bisphosphate by phosphofructokinase. This is an irreversible and a regulatory step, rate limiting step of this glycolysis. It is, which means the essential step. This step only start the actual actual glycolysis. The next step, splitting phase. The fourth step, splitting means the fructose 1,6-bisphosphate is cleaved into two various chemicals. The 1,6-bisphosphate is splitted into two 3-carbon compounds. With the fructose has 6 carbon that is divided into 3. The first compound is known as glyceraldehyde 3-phosphate and later on as dihydroxyacetone phosphate. This step is carried out by the enzyme aldolase, or other name is fructose 1,6-bisphosphate aldolase. So the splitting step is carried out by the aldolase enzyme. Then fifth step, the enzyme phosphotriose isomerase catalyze an irreversible interconversion of glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. So interconversion between them. In this, glyceraldehyde 3-phosphate is converted into dihydroxyacetone phosphate, and the dihydroxyacetone phosphate is converted into glyceraldehyde 3-phosphate. Here, from this step, until this step, we have one molecule, but then after this step, we get the two molecules of glyceraldehyde 3-phosphate. So two molecules of glyceraldehyde 3-phosphate are obtained from the one molecule of glucose. Next, final phase, energy generation phase. The first phase, energy is invested, that means the energy needed. Next phase, the splitting phase, the chemical is divided. Next, third phase, energy generation phase. In this phase, the energy is synthesized. For this, the glyceraldehyde 3-phosphate dehydrogenase enzyme converts glyceraldehyde 3-phosphate to 1,3-bisphosphoglycerate. This is the important step, the formation of NADH plus H plus and high energy compound. So next, the enzyme phosphoglycerate kinase act on 1,3-bisphosphoglycerate, resulting in the synthesis of ATP and the formation of 3-phosphoglycerate. This is the substrate level phosphorylation step. So ATP is synthesized in this. Then next step is the 3-phosphoglycerate is converted to 2-phosphoglycerate by phosphoglycerate mutase. This is also the isomerization step. The ninth step, the high energy compound phosphoenol pyruvate is generated from 2-phosphoglycerol by the enzyme enolase. The enzyme require Mg2 plus or Mn2 plus. These are the cofactor. And then final step, the enzyme pyruvate kinase catalyze the transfer of high energy phosphate from phosphoenol pyruvate to ADP and produce a ATP, and this step is the essential step. So this step also the irreversible step. Finally, we get the pyruvate from the phosphoenol pyruvate. The final product is pyruvate. So this is the aerobic glycolysis. But in anaerobic glycolysis, that but in anaerobic glycolysis, the lactate dehydrogenase enzyme dehydrate the pyruvate and produce lactate. So this is the final product of the anaerobic glycolysis. So this is thank you all.